Common.
Yeah.
What shows isn't like everything?
Because of dominance.
Dominance.
Because of the fact that you cannot, why can't you distinguish these two? Because if you happen to be a pure round versus a mix of round and wrinkled, you cannot distinguish them because round dominates over wrinkledness. And that is dominance, that's called dominance. So now let's go back and look at the laws that Mendel came up with. Law number 1 is when this mating process happens. So first of all, every individual, everybody has 2 copies of what we call the allele, and these 2 copies are called the gene. Yes.
That's the first one?
No, I'm just saying, that's not the first law. That's only part of the first law. First law says every individual has 2 copies of a trait, and that's called a gene nowadays. So everybody has a gene for their different traits. And when the mating process happens, there is this process of segregation. Segregation means what? Does anybody know what segregation means?
Like divided.
Separation. Separation. So what happened was you had 2 copies of this, and these got segregated, and you got either one or the other. Like this is one of the parents. Because at the end of the day, the child after mating also has to end up with 2 copies, right? Everybody has to have 2 copies. So this is the thing in segregation. From one parent, you are segregating, you're separating these 2, and by chance you get either this or this. And from the other parent, exactly the same thing happens. That's called the law of segregation. Then there is the law of independence. The law of independence says that if you have 2 traits, they combine independent of each other. Okay? And then third one, some people call it a law, some people don't call it a law, but this concept of dominance. Right? The concept of dominance is if you have 2 copies of a trait which are different from each other, one trait will dominate over the other. Now, Mendel's laws apply to pea plants very nicely. It doesn't apply in the real world to everything. And as we go along and understand, we go in more of the depth of how the molecular or the cellular basis of these are, like why, like how these things are actually put into the cells and how the whole process of reproduction happens. You'll better understand. You'll better understand why, in some cases, Mendel's laws doesn't quite work. But like, not all the time dominance shows, not all the time independence shows. But at least this is radically different from how people thought of heredity before Mendel came. People always thought that if you have 2 traits and you mix them up together, you will get something which is somewhere in the middle. So tall and short would become like sort of medium, and round and wrinkled would become like a smoother wrinkled. But Mendel showed that that was not the case for peas. and he came up with all this. Okay? So now what I'm going to do is, I'm going to just show you some problems.
Are we good on time? Are we good on time for all of you? 10 minutes?
I don't know if there is another class you have to go to.
No, yeah.
So,
I have to go somewhere.
So you have to leave right now?
No, I can leave in like
10 minutes?
okay. So let me at least show you some problems.
We
And
need
then
the
what I want
lights
to do is I
off.
want to
We need to dim the lights. Thanks.
I'll
Thanks.
actually work with the other coaches and come up with a problem set that you can do at home, which will help you kind of refresh this. And what I did is in the Google Drive, I actually
Yeah, it's all there in the Google Drive.
I actually put a bunch of material.
Is it on that side? Yeah. Thanks.
Thanks for putting all the material, uploading all the material.
Yeah,
In
it
fact,
was
I'm
very
using-
helpful.
I'm using her slides. She's
I don't know how long is this, how much time do we have here? It's 1 hour, 12 minutes. We are good?
yeah, it's good.
Good for 10
No
minutes.
problem.
Thanks.
So this is basically
I
the.
need to have, I can't get this. Do you need this to be on the video? Because you want to explain this?
No, I won't. I'll just go over it. So just keep the
Bec-
video on.
Because we can't see it on this with the lights on. Like video camera, it's blurred.
Oh, that's okay.
Because this is going to go on the.
Yeah, but at least just keep recording. That's it.
Yeah, but whoever, they can't see it from
That's
this.
fine.
Okay.
They will look at
Get
the
it
slides.
anyway. Okay.
Yeah. So if you go through the slides, this is the lesson 1 slides. It basically talks about Mendel, and this is his garden, and it talks about like some of the stuff that he did, and I hope now if you read these slides, it'll make a lot more sense. So look, this is what he did. He crossed the pea plants, and for the different traits, and this was all statistical, and these are the different. So he took tall and short and
Action.
did all this. So he called character corresponds to like a gene, and trait okay. Character is basically the attribute, and trait, so character corresponds to the gene, and trait in this case corresponds to the allele. And then he talks about the dominant and recessive. Does it make sense? And he talks about the experiments that we talked about. And then here are some new terms.
Mosaic.
And by the way, all these terms are important. You need to memorize them. They, in Science Olympiad, I looked at the questions. They quiz you, assuming you know these terms. So phenotype and genotype we covered. Homozygous and heterozygous we did not cover. Homozygous is pure. The TT would be homozygous, and heterozygous is a mix. So TT and small s is homozygous. Heterozygous is where it's not that. And then in the notation, just because this was for a lesson, I said T and S. The normal way of representing this is, even though T and S are like one is tall, one is short, you just pick a letter. It doesn't matter. It could be T, it could be A. Usually people pick the letter for the dominant one and make it capital. And then you don't write short, you write small t. So, using that same logic, what would this W be? Small r. Good. So, and what this shows is this r, this capital thing, this is the dominant one, this little one, even though it's not roundness, it is wrinkledness, it is the recessive one. So this one usually tallies up with the dominant whatever character is there. This one doesn't tally up with that. Otherwise you would have to write W. So that's the notation you'd see all over. Okay, and that's what the lowercase and uppercase is. And this you'll see the first generation is called F1, the second generation is called F2, third generation is called F3. And by the way, you can go and take the second generation. We didn't go beyond the second generation, right? You can go beyond the second generation, see what happens. That will be a good exercise for you to try out at home. Okay? And phenotype versus genotype. Does everybody understand it? You can have different genotypes, but it might show up as the same. Always remember, the tall tree may be a tall tree, a real true-bred tall tree, or a tall tree which looks tall, but might produce some short trees. So it's not pure. So that's the difference between genotype and phenotype. Okay, and they talk about this, and dominant and recessive. You know what alleles are, right? Alleles are the individual components. That one T is an allele. So in this thing, what are the two different alleles you have? In this whatever we talked about, what are the two different alleles for the height of a tree? Yeah, capital T and small case t. Exactly. Okay, so you kind of know this, what you observed, we went over what his rationale was, and we sort of tried to cover that, why he thought the way he thought. And this, I think you now understand what I said. Okay. Gametogenesis, this is a fancy name for the fact that segregation is happening. Basically, you have two copies and either one of them gets picked up. That's the law of segregation, right? That's called gametogenesis. And fertilization is when these two from the two different parents come together and become like the child individual. Okay? Okay, so this is, we kind of, okay, we didn't do this explicitly, and we'll start with this Punnett square in the next lesson. But Punnett square is really a fancy way of, not a fancy way, actually it's a pretty systematic way of representing what we just did it in this table. It's done as a square, so Punnett square is pretty straightforward. So instead of doing this combination like the way we did, T, so you draw a square. So this is a, one parent can give one T or the other one can be T. This is generation 1. And what does the other parent give?
R.
No. Same trait.
Oh,
It's the
SS.
same trait. It's
Small
not
t,
S, it's
small
now
t.
small t. That notation that I was using earlier was just to make things simpler for you to understand. Going forward, we'll not use two different letters. It will be the same letter. And Punnett square, all it says is, if I got this from here and this from here, I would get a TT. If I got this from here, I'll get a TT. If I get this from here, TT. And it's not very, like, it just is another way of representing what we just said, right? But this is sort of useful when you try to do not just T, but you put other characteristics in here. Like if you want to do a Punnett square with two characteristics, it becomes a lot more interesting. And also when you do second generation, it becomes interesting, right? So this is called a Punnett square. There are a lot of problems in Science Olympiad which have to do with Punnett squares. Okay, so you know all this now. You understand this? You understand?
Yeah.
This stuff? You'll be able to go through these slides and understand, right? And this is the law of segregation. I think we covered this. Monohybrid cross. Monohybrid is when we are crossing just for one trait. The first experiment, the tall and the short, that's called monohybrid. Dihybrid means I have two traits. And Science Olympiad questions will be usually on dihybrid. Okay. Test cross is when you don't know anything about a plant, what do you do? So you can see. This is the law of independent assortment. The law of independence that I said. The two characteristics are not tied with each other. Okay? Dihybrid cross, this is with two traits, not one trait. Okay. Dependent segregation, right now we won't cover this. This is when they are not independent. Okay. So Mendelian genetics, you can do this. This is basically that table, it's drawn as a Punnett square. And they'll always tell you which one is dominant. By the capital letter, you'll know which one is dominant. Okay. 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 We'll cover some of this in subsequent classes, but I think this is pretty good for today. For now, we can. I didn't cover the parts that are there in this slide. So one quick thing I want to show you. I'll give you some problems, and I'm going to go over. So if you go to your Google Drive, you'll see this is the folder, the Science Olympiad folder. If you go in there, you'll see assignments, class presentation, and the designer, genes C and heredity is taken from the CD. So right now, you don't really need to go over this, but in assignments, I put a bunch of stuff, and I need to sit with the other coaches and figure out how to do this. But at least this one, problems_lecture 1, this is the one I think I looked at it and I have. Okay, the third.
You're out of battery here.
Okay. The problems of yeah.
I have to go now.
Yes, sure. So I'll send out in email these
Yeah, I think that we can do it.
these things, what needs to be done. And I need to talk with the coaches also. But you might want to try these problems.
Okay.
they are fairly, if you understood this, I think you should be able to think, and again, feel free to ask the other